Hi, everyone. My name is Kenny Coogan, and I'm the Education Director for the International Carnivorous Plant Society. For a couple of years now, we've been offering monthly webinars, and I'm very excited to introduce Jason all the way from Taiwan to talk about a fungus that helps prey digestion in carnivorous plants. If you're attending this live, feel free to type in questions into the chat box below. And when Jason is done, I will read him all of the good questions. And thank you, Jason, for agreeing to do this presentation for us.
Thank you, Kenny, and thanks for the invitation. It's a pleasure to talk about our research or recent findings entitled "An acidophilic fungus promotes prey digestion in a carnivorous plant." So just a quick introduction about my lab. So I'm located in the Biodiversity Research Center of Academia Sinica in Taiwan. And my lab has focused on exploring the genetic and molecular diversity of East Asia's unique flora and fungi. Taiwan is actually an island with quite extraordinary biodiversity, and so we characterize some of the species diversity. Once they are quantified, then we go on and attempt to investigate the evolution and mechanisms behind this diversity. So to define one such environment, here I'm just going to show you a short video of a forest that we frequently went for sampling. So this is a typical what a broadleaf forest consists predominantly Fugacia species look like. So oak trees, beech, etc., etc. Right. So and here's a photo of a typical beautiful broadleaf forest in Taiwan. In the most basic sense, my lab wished to know if we could interpret the whole picture literally. You know, what trees and fungi are present, how are they distributed, their fitness, interactions, and how they really evolve with each other in this environment at a microscopic level. So if I break it down to what I really know about this photo, it will look something like this. You know, a vast unpainted canvas awaiting exploration. And it's really because in how scientific research is conducted. You know, much of the understandings comes from very few organisms. You know, they are typically very human-oriented. You know, disease, pathogens, agriculture, or human themselves, or the so-called research model organisms. So my lab's strategy is a bold one. We just try to tap into the unknown domains. So our exploration of these fascinating ecosystems has led us to study quite unique species, such as bioluminescent fungi, the stalk camphor tree, and now the carnivorous sundew plant, Drosera spatulata, and its fungal partner. So the research journey really began when the PhD student Pei Feng Sun asked a simple yet quite profound question. You know, can microorganisms influence the carnivorous mechanisms of plants? I wouldn't need to mention too much in the audiences here, but you know, carnivorous plants have long captured the imagination of scientists. a pleasant historical background, and much inspirations come from Charles Darwin. Darwin is probably one of the most influential scientists in history, known probably by most people about his original species work. He was actually very captivated by carnivorous plants in his late days. Darwin's favorite organisms were Drosera, the sundews. He meticulously observed how the plant's sticky mucilage trapped and digested insects, and he described his experiments in his book Insectivorous Plants, published in 1875. As a fungal geneticist or slash evolutionary biologist, I absolutely shared his thoughts in his extensive correspondence with fellow naturalist Charles Lyell. He said, and I quote, I will and must finish my Drosera manuscript, which will take me a week. For at this present moment, I care more about Drosera than the origin of all the species in the world. This statement really underscores just how enthralled he was by these remarkable plants. And also, no matter what career stage you are in, you always have a deadline to finish manuscripts as scientists. Since then, carnivorous plants have always been of broad interest and are categorized by their trap mechanisms. Taiwan is home to 3 native Drosera species. Drosera spatulata, in particular, is distributed in northern Taiwan, has mucilage secreted by stalked glands, which traps and digests prey. And not just a quick video of how an ant was fed and trapped by the mucilage in the lab. And again, for the audiences here today, I wouldn't need to mention that the leaf will reopen once the digestion process is completed. The mucilage often exposed to environmental influences, right? For example, just the raindrops, will pretty much perturb the mucilage. So it makes us wonder what microorganisms live in it, and do they play a role in digestion? We made the following hypotheses. We hypothesized that there are various microorganisms present in the mucilage of Drosera. The microbial community is likely to be quite dynamic and noisy because of external influences. We further hypothesized that some of these microorganisms positively enhance the sundew's digestion process. So here we see a typical Drosera habitat in Taiwan, with sundew plants growing on the lit cliff surface along mountain trails in the dense forest. These environments are incredibly unique or harsh, perhaps, with fluctuating moisture levels, and the steep cliffs create microhabitats where nutrients availability is low, making it a perfect setting for carnivorous plants like Drosera to thrive. To investigate the microbial community, we sampled mucilage from 3 sites in northern Taiwan and collected controls from nearby non-carnivorous plants. We then extracted the DNA for metabarcoding of both bacterial 16S and fungal ITS regions. So our analysis revealed overlapping bacterial communities between the Drosera mucilage, as shown in the red dots, and the surrounding plant surfaces, as shown in the green dots, indicating there's really no significant overall bacterial differentiation between the mucilage and the surrounding environment. However, the fungal communities shows a clear separation, as shown in the horizontal axis, right, indicating significant differentiation in the fungal community. The next was very straightforward. When we investigated the major microbial taxa throughout all samples, we found that the mucilage was dominated by Aquodontium crateriforme. As you can see in the box plot here, this has an average around 50% of relative abundance per mucilage sample. Just wondering, can you guys see my mouse cursor?
Yes, we can.
Okay, that's good. Let me just try to use this. Yeah, okay. So we next sought to confirm if Aquodontium consistently remained the most abundant fungal species in Drosera mucilage over time. Now, sampled from various two sites in northern Taiwan over 18 months, we observed its dominance even through seasonal changes, right? So you see a big red color here, while the abundance dipped slightly through the hotter summer months. Aquodontium still maintained a significant presence. And again, to just show that our findings wasn't restricted to a few locations, we expanded our geographic sampling across northern Taiwan, and it shows again Aquodontium is the most dominant fungus in the microbial community of the mucilage. So this consistency suggests that Drosera Aquodontium relationship is not only temporarily stable but also geographically widespread. At this point, I'd like to share a story about openness, right? When we submitted our work first as a preprint, so the preprint is a paper, considered a manuscript that hasn't been reviewed yet. So it documents the observation of dominant fungus in Drosera. So a PhD student, Brandon Shaw from University of Loughborough in the UK, later sent an email mentioning that the Aquodontium was also present in 2/3 of his samples across multiple species. This resulted in collaboration with meta-barcoding data available in multiple Drosera species, including the Darwin's favorite, Drosera rondifolia. And we discovered Aquodontium quadriforme. Aquodontium is also a dominant fungus in multiple Drosera species outside of Taiwan. However, there are some variations. In the UK and USA samples, Drosera rondifolia showed a bimodal distribution. Certain samples had really high abundance, even higher than the average ones in Taiwan, while the others had lower levels. And we also found that in the case of Drosera intermedia, it contains very few Aquodontium reeds. So the variability highlights the influence of possibly local environmental conditions on the fungus prevalence. Yet the widespread presence across continents reinforce this plant-fungus symbiotic relationship is ancient and possibly co-evolved. So here we use scanning electron microscope SEM image of Drosera glands grown under sterile conditions in the lab. This will establish tissue culture from seeds collected from the sampling sites. Notice how the glands appear very smooth and shiny, completely free of fungal hyphae. This also suggests that Aquodontium, the fungus, does not vertically transmit from plant to plant through seeds, but instead colonizes the mucilage from environment after germination. In contrast, this SEM image above shows that Drosera gland inoculated with Aquodontium. The fungus has grown extensively with the hyphae adhering directly to the gland surface. This also meant that we cannot conduct the experiments in the fungus and the plant separately or when added together bringing possibility to various experimental designs. Here's a close-up of Aquodontium on the Drosera gland. The image highlights conidial spores, some with attached conidia and others with detached conidia, showcasing the reproductive structure of a fungus, and really confirms that all of the life cycle, all parts of the life cycle can be of the fungus can be completed on the glands, really showcasing the intimate relationship between the plant and the fungus. So we have established the pure culture of Aquodontium. It looks like this on the media petri dish. The first logical step is to delve into the basic biology, right? So as we really were a microbiology lab at the beginning. So we conducted controlled growth experiment. We adjusted the pH of culture media and observed that Aquodontium thrived in these acidic conditions at pH mimicking the acidic mucilage. Comparatively, the different fungus, Forma herbaceae, which we also isolated but as a major taxa from the surrounding environment, prefer more neutral pH and did not grow well under acidic conditions. This highlights the acidic nature of acidophilic nature of Aquodontium, perfectly adapt to the extreme environment of Drosera mucilage. Next, we have sequenced the genome of the fungus, and our phylogenetic analysis have placed it with a clade of non-extremophilic fungi, including the well-known acidophilic Acidomyces, Richardonensis, and Neohotia acidophila. These groups of fungi are usually found in very, very extreme environments like the Arctic or very, very high salinity environments. So we discovered that the genomes of Aquodontium share several unique adaptations that enable it to thrive in the harsh acidic environment of the Drosera mucilage. One striking feature when we compared the gene families dynamics, which and found that Aquodontium exhibits extensive gene loss compared to sister taxa, as depicted by the larger size of the blue circle here. So when looking into the which families were lost on the right, we found that these families associated with plant pathogenicity, for instance, genes that encode cell wall degrading enzymes commonly used by pathogenic fungi to invade host tissues, are largely reduced or absent. This reduction really mirrors what we see in other symbiotic fungi studies, such as actomycorrhizal species, which have similarly lost many of these pathogenic traits as they evolved more cooperative relationship with their plant host. So kind of recap our findings and original hypothesis. Oh yes, that is our microorganisms. But in contrast to our original predictions, there's actually a dominant fungus, Aquodontium quaterniforme, adhering to the mucilage gland of Drosera. It grows optimally at acidic conditions, thriving on Drosera glands. Is one of the extremophilic fungi and genomic signatures typical of symbiotic fungus. So this left us with the final hypothesis, right? Is the sum of these microorganisms, or in the case here, Aquodontium, positively enhance sundews digestion process? Or in a way, we wish to test a new paradigm, whether the digestion process in carnivorous plants is a holobiont process, i.e., a mix of organisms, right, involving the plant host and the microorganisms, and that Aquodontium is actually part of a process contributing to the overall digestion. That also means we need to start inoculation, right, in the lab to test responses of Drosera with or without fungus. In fact, loads of inoculation. This is just a typical image of the amount of samples that the student need to inoculate for one particular experiment. When trying to assess what kind of substrate we needed to use, here's a fun historical anecdote from Darwin's era about carnivorous plants that made it into the Nature journal in 1874. Beef was a great success, but not cheese. So the moral of the story is, if you really want to get into high impact scientific journals, just feed your plant a steak a day and hope for the best. But it's really not your typical plant experiment. Joking aside, the first key assay we designed was a reopening time assay, where we exposed the Drosera traps to different stimuli. We either gently touched the traps or added a variety of substrates. Non-nutritive controls like wood powder and shrimp shell, or ant powder, which is a natural prey source. We then recorded how long it took for the traps to reopen, comparing the control plants to those inoculated with different microbial treatments. First, the non-nutritive controls or touch resulted in reopening time just under 20 hours. So they acted as a perturbation of external stimuli that will force alarms, i.e. no food, not food. This is a note that if you think about it in the ecological side of things, it's actually very energy consuming and a very expensive process for the plant if the plant recognizes the wrong signal or wrong food. So, and the treatment included a sterile control group with no added microbes, which resulted in reopening just under 100 hours. Then microbiota from wild Drosera musage, and the microbiota or the mix of microbes from the surfaces of adjacent plants in the wild. Meaning that probably just contain different combinations of microorganisms. And already we found a significant reduction in the reopening time that was observed. And finally, and perhaps very excitingly, inoculating with pure aquadontium resulted in the most significant reduction in reopening time, meaning that it's almost a quarter of time was saved in the reopening or digestion process. Let's take a step back from the excitement. One counter argument to our findings could be that the faster reopening time of Drosera traps are really not the direct effect by the aquadontium, but instead reflect a so-called primed physiological state of the plant. In other words, the presence of microbes may induce a form of defense priming, where the sundew plant cells become more responsive to external stimuli, even at very low levels, leading to more rapid reactions, including digestion. So we need to design another assay to directly assess aquadontium presence. So one evidence in why we believed aquadontium may act directly as a digestion partner in Drosera musage is from the growth profiling. So aquadontium exhibits significantly enhanced growth in the ant supplemented media compared to the standard media. So by day 10, HIFO coverage in the ant-enriched media was nearly double that of the control, suggesting that the fungus thrives in a nutrient-richer condition, similar to those provided by the breakdown of insect prey in Drosera mucilage. So we designed another digestion assay. So by mixing biotin labeled bovine serum albumin (BSA), so this is a common source of protein, with the collected mucilage from plants with or without different microbes inoculated, we wish to quantify the efficiency of protein degradation after 24 hours. On the y-axis, we have a percentage of undigested BSA remaining, and on the x-axis, the time point of 16 and 24 hours after mixing. And we see here that there were no significant difference in the 3 treatments, sterile plants versus the 2 wild microbiota. Strikingly, when inoculated with aquodontium as depicted in the red box, there was a significant reduction in undigested BSA at both time points, indicating that this fungus substantially enhances the protein digestion process in Drosera mucilage. At this point, our findings illustrate the key insights into Drosera aquodontium relationship. First, it significantly enhances the digestion efficiency of Drosera, possibly through priming, but also demonstrating aquodontium is not really just a passive player. It actively benefits from the prey, insect prey, indicating there's a mutualistic relationship where both organisms capitalize on the nutrient resources available. In the final setup, we aimed to investigate the transcription dynamics of Drosera and aquodontium Holobiont under various conditions related to digestion. We designed 3 experimental scenarios in addition to the baseline control, just the plant only or the fungus only, in attempt to dissect the molecular dialogue between the plant and fungus during prey digestion, and really uncover how this partnership enhances the overall digestion process. So we have the coexistence condition phase, where Drosera is inoculated with fungus, but without any ant prey. So this really setup allows us to examine the interaction between the plant and fungus. Second, we explore the digestion condition where Drosera or fungus alone are provided with ant prey, but without the presence of either partner. So this scenario reveals, how the plant or fungus transcriptional response to digestion alone. And finally, we have the holobiont digestion, where Drosera is inoculated with aquodontium and supplemented with ant prey. So this really comprehensive setup, hopefully to enables us to understand how the combined presence of prey and fungus influence gene expression in both partners. Okay, so by comparing the transcriptome response of coexistence and digestion process in a plant, approximately 60% of differentially expressed genes in the digestion phase was also significantly expressed in the coexistence phase, and in the same trend. So if it's upregulated in digestion, is also upregulated in coexistence. Let's reveal that digestion-related genes in Drosera are not entirely new innovations, but have been co-opted from pre-existing gene families related to plant microbial interactions and defense. This observation is quite consistent to previous genomic studies of carnivorous plants, that gene families involved in defense were duplicated and evolved to gain the digestion ability. And in the holobiont digestion transcriptome, we found that more than half of the upregulated differentially expressed genes in both the plant and fungus show multiple or additive effect when both the partner and the digestion stimuli were present. This really just so when I say additive, meaning when both stimuli are present, the gene is upregulated even further or is being amplified even further in a synergistic manner. And when we talk about carnivorous plants, we have to talk about nitrogen assimilation. And one key observation is the active exchange and utilization of ammonium between Drosera and Aquodontium. And the upregulation of all these nitrogen utilizing pathways were observed already in the coexistence phase before the insects is introduced. And it really highlights the recognition of partners, possibly at the prime state in the plants, right? So, and when it has fungus covered on the glands, it's already prepared, for the next insect to arrive. So we also discovered many proteases and peptidases which are involved in the breakdown of insects. And in the example here, which is the asparagine synthetase, crucial for nitrogen storage and transport in plants, was specifically upregulated and amplified even further only during pre-digestion with the fungus present. So it really clearly shows, the marked increase in asparagine synthetase activity, and it really underlines kind of a strategic nitrogen assimilation facilitated by the fungal partner, which helps maximize nutrient retention and utilization. So all these combinations of comparisons allows us to dissect and perhaps provide a comprehensive view of transcriptome dynamics occurring during holobiont digestion in nature. On the right of the plot, you see the digestion phase, but without partner, where peptidases and transporters are upregulated in either species. On the left, you see the coexistence phase. Again, the peptidases, ion transporters, and some of the secondary metabolic processes are overlapped with the genes in the digestion phase. And so in a sense, they are in a primed state. And finally, in the middle, in the holobiont digestion phase, both organisms coordinate to break down prey efficiently, with Drosera enhancing even more of different diversities of transporter activities and asparagine synthetase, while Aquodontium contribute to really much more peptidases like cell-donicin and aspartic enzymes to further break down the temporarily nutrient-rich environment. To summarize, some deals employed a shared gene pool for digestion and coexistence processes, and confirmed that the genes that related to digestion were indeed co-opted from genes previously involved in interactions of defense. And colonization of microorganisms, not just Aquodontium, but any microorganisms, consists of biotic stimuli that enable the plant to undergo transcriptomic changes before the prey is even introduced at the prime state. But the case of here is quite special. Holobiont expression patterns during prey digestion really further reveal the synergistic effect in several gene families, including the fungal aspartic and cell-donicin peptidases, facilitating prey digestion in leaves. Kind of looking forward now, Our next steps will involve exploring nitrogen assimilation pathways in Drosera more deeply. As shown here, instead of relying on soil uptake, Drosera utilizes prey-derived nitrogen with key enzymes we already know are upregulated during digestion. An exciting area, I think, will be the potential role of nitrogen fixing bacteria, perhaps also present in the mucilage and contribute to the process. And by digging into our previous bacterial metabarcoding data, we actually found there's an enrichment of numerous Burkholderia taxa. They are not dominant, but they are present in various low percentages. And this one is interesting, Burkholderia, and in the past literature actually revealed that it's a genus in plants associated nitrogen fixers. So perhaps this is something of a tri-part relationship. Another one of recent breakthrough in our research have been the assembly and the genome sequencing of tetraploid Drosera spatulata. This is necessary because previous sequencing efforts was from a New Zealand individual which was diploid, whereas the rest of the global populations, including our subjects, are tetraploid. So the updated assembly has an impressive size of 1.17 gigabase with high contiguity, and hopefully we can compare the genomes of the diploid and the tetraploid to open new avenues for really understanding the genetic complexity and adaptive evolution in these plants. And just to conclude, our study has redefined the symbiotic interaction between Drosera and Aquodontium, and really provides new insights into botanical carnivory that build upon Darwin's legacy. We were the first that demonstrated that a microbial partner not just enhances the carnivorous plant digestion process, but also influences nitrogen metabolism with evidence of specific protein degradation pathways activated. The Drosera Aquodontium system and potentially involving bacteria offers a quite nice laboratory model for detangling the very complex plant microbial relationship, and hopefully is paving the way for more exciting research results. So on that note, that concludes the end of my talk. Just a final slide that I would like to acknowledge in the invaluable contribution of my incredible research team. Special thanks to Pei Feng, my former PhD students, and actually all our collaborators, including Brandon, who kept in touch with us, Yinlan, Roland, and Jimmy. I'm actually grateful. I need to say here, I'm grateful for the support from my own institute, Academia Sinica, as well as NBO and NSTC. So thank you for your attention today, and I'm happy to take any questions you have.
Thank you so much, Jason. And
Yeah.
I'm really excited to share this presentation because when we posted it on our social media, we had a lot of interest. I'm just going to talk a little bit about the ICPS. Jason, if you go to the chat, there's a couple of general questions for you, which I'll read out in a moment. But Jason just did a wonderful webinar for the International Carnivorous Plant Society, and I am the education director. I'm a board member. And our society consists of about 1,400 paid members, and those memberships help keep our 2 websites running, our seed bank operational, and helps with conservation and education initiatives. Coming up in 2024, we have a couple more webinars, and I reached out to Brandon Shaw and Jason separately, but Brandon signed up for a webinar next week, which is going to complement this wonderful webinar. So he's going to be talking about deadly symbiosis, and then we have a couple more coming up. And then in 2025, we have 2 more webinars in January and February. So you can find out and register for all of these free webinars by going to our Facebook page or by going to our website, which I'm going to share in a minute. If you are a researcher or a grower, you can contact me at kenny@carnivorousplants.org, and I can set you up for presenting for our organization. A couple of years ago, we made some animated videos to talk about what is a carnivorous plant, how to grow them indoors, how to grow them outdoors, and how to feed Venus flytraps. You can check these out on our social media pages, including our YouTube page. And the first Wednesday of May every year is World Carnivorous Plant Day, so that means that the date changes, but it's always the first Wednesday of May. You can go back to the first couple of years, and you can see over 20 videos each year about growing carnivorous plants, about carnivorous plant research, and you get a whole lot of educational content. To become a member, you can go to the icps.clubexpress.com. And like I mentioned, your paid memberships keeps us going. We have been around for 52 years. And one of the biggest benefits that our members share is that we have a seed bank that can ship internationally. And some of the seed packets come with instructions like they need to be cold stratified. Others you can just surface sow. And we can ship to many countries. You can contact our seed bank manager for more information. But you do need to be a member of the ICPS to get access to our seed bank. And then the other really big benefit of being a member is you get a beautiful quarterly newsletter. So Jason just did this presentation in November, but yesterday I received my winter December 2024 issue of the Carnivorous Plant newsletter. And we kind of marked the age of the ICPS with our Carnivorous Plant newsletter, which has been going on since 1972. If you become a member, you get this beautiful full-colored newsletter. But what I think is more important is you get access to the last 52 years of newsletters, which have scientific report, field trips, cultivar registration, conference updates, lots of good stuff there. Every August, we have a Carnivores in the Classroom grant for teachers who teach kindergarten through 12th grade. To learn more about that, you can go to our website. And to win $150 of carnivorous plants shipped to your classroom, teachers or students have to create a lesson. And if you go to our website, you will be able to download all of those lessons for free. So if you need to keep your kids busy during a spring break, you can use these, or if you're a teacher, you can use them. And these are our sponsors for 2024. We're very happy with them. And most of them are nurseries and individuals who wanted to donate plants. But we did have a new partner this year, Carnivorous, who makes 3D printed carnivorous plants. They gave us 5 instructions for free. So if you go to our website, and if you have access to a 3D printer, you can print your own carnivorous plant. And I hope you can see my awesome t-shirt. It has Sarracenia purpurea on it. If you go to this website, you can purchase carnivorous plant t-shirts, hoodies, tank tops, tote bags, and all proceeds go to our education and conservation initiatives. If you search into our newsletter, you can see what projects we are funding regarding conservation. We have a conference, we have an in-person conference every other year. 2025, we're taking off, but in 2026, we're going to be in the Americas. We are probably going to be in North America or Central America. We will be announcing that in a couple of months. All right, thank you, Jason, for that commercial. So Jason, I'm going to ask you a couple of basic general questions, and then once I post this, you'll probably get some more specific questions. Jason, what advice do you have for young people who want to get into carnivorous plant research?
That's a good question, Kenny. I think that there's always carnivorous plant enthusiasts out there, and there's a lot of plant biology lab, and if you just talk to the I would say professors who already have a research line going on, but not carnivorous plants, you can really take you can really approach the professor, say, look, this is I'm interested in carnivorous plants, and I'm also interested in these aspects which you have done. Can I do it in my species per se? And I think most professors will say yes. I mean, this is the way I approached or when I started this particular research. My student came to me saying, you work on fungi, you are a microbiology lab, but I really like carnivorous plants. Can I work on this particular topic? And so I would say this is the advice I would give. Yeah.
Yeah, that's a great answer, because there are a lot of carnivorous plant researchers, but there's probably always room for more. And if a local university isn't researching that, but they're researching something in a similar topic, that's a good gateway. This next question kind of follows that. The people who work in your lab, would you say they are more interested in carnivorous plants, or are they more curious in ecology and symbiosis and biology, and then they're just using the carnivorous plants as a model?
I would say the latter. So they are just generally fascinated with biology and ecology in general. But we are starting to develop a research line that specifically will tailor to carnivorous plants. So maybe there are hidden players or students who just wish to go for it. Yeah.
Can you give us what percentage of your work includes this symbiosis with fungi? Or can you tell us, like, what are some of your upcoming projects, even if they're not related to carnivorous plants?
Okay, so I work on all kinds of fungi, and I do mainly work on yeast. So for instance, the baker's yeast, right? So one of my main project was to see the ecology of yeast, in the sense that we know yeast as probably Saccharomyces, where you use the yeast to brew wines or make bread. But we were more interested in what it does in its natural habitat in the forest, right? So and yeah, just anything that is fundamentally interesting and important in the terrestrial system, terrestrial ecosystem like the forest. I've worked on bioluminescent fungi as well. So that are just fascinating group of organisms. So yeah.
Daniel asks, did you test any Drosera you had in the lab prior to starting these experiments to see if they had presence of the fungus?
Yes, we did, and that's why I mentioned previously in the slides that they were actually very um, free of the fungus, and we need to test that as well because we need to set up experiments comparing the responses with and without the fungus. Yeah.
Daniel came in a little late, but they also asked, did you do any tests to see if you could move the fungus from a plant with the fungus to a plant without the fungus?
Sorry, what was the question again?
Were you able to transfer the fungus to a different Drosera or that didn't
Oh
have
yeah.
it initially?
So we can grow the fungus separately. And we can, we grow the plant and the fungus separately, right? So once we have the fungal culture, we can always inoculate onto different Drosera plants, right? And the plants we grow are sterile, i.e. they are devoid of the fungus in the usage, right? And we can.
Are you doing different Drosera species?
We plan to, but for now, because that's Drosera spatulata is actually a cosmopolitan species that's present across, or at least this part of the world, and so we plan to see if there are individual differences uh, response um, to different strains of a fungus. So one thing we can do is we collect Drosera in Japan, and we see if the fungus again if the sundew content in Japan contains Acodontium, and is the genetics of the fungus in Japan different to the fungus genetics in Taiwan. And then we can do the different combinations to see the responses. Yeah.
Okay, Jason, the last question. Anna says, do you plan to expand research on the topic of symbiosis between fungi and other carnivorous plants other than the genus Drosera?
Yes, definitely. So one thing I didn't mention in my talk was that we actually downloaded all the past literatures and try to search for the presence of this particular fungus in other carnivorous plants. So there were some studies that have been done looking at different carnivorous plants and with obvious choice of pitcher plants, right? And um, there is actually the presence of this fungus, but at very low abundance. So, and my hypothesis was that we need to redo the experiment. So, for instance, as you can see from the microscopic pictures, it's really adhering to the gland surfaces, the fungus hyphae, right? So, and how the previous work were conducted, they simply collected the digestive liquid of the pitcher plant. So if the fungus is actually growing on the inside the surface or above the surface of the pitcher
liquid.
plant, and then we wouldn't able to detect it. So one thing to redo the experiment would be to scrap the surface or the inner surfaces of the pitcher plant to see if there's actually fungus present. So I think some of the experimental methodologies need to be revisited to confirm some of these speculations. Yeah.
All right, thank you so much, Jason, for a great talk. And thank you everyone who attended this live. And we hope to see you in a future webinar. Thanks, Jason.
Okay. Thank you very much.
It's not a surprise that gardeners, educators, and scientists are fascinated by these unique plants. The International Carnivorous Plant Society, or ICPS, not only love these plants, but welcomes growers just getting started all the way through professional scientists. The ICPS even started an annual World Carnivorous Plant Day to celebrate them. The free online event is held the first Wednesday of May. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.